We all have that problem. The problem where you were just cutting your apples for your afternoon snack, and just when you went and get a glass of water, and when you come back to it, you realize the apple has turned brown. This is what we call enzymatic browning. Fruits and vegetables such as apples and carrots can be oxidized when it is left out and exposed to the air for a while, leaving a brown and dark pigment appearance to it. And the longer it is, the darker they become. And this happens because apples and carrots contains two enzymes called polyphenol oxidase, PPO, and peroxidase, POD. The common solution that everyone knows to this problem is by salting them, applying lemon juice, or boiling them. Salt, which binds with water, can lower down the water activity of the solution, and lemon juice changes the pH value of the solution and the antioxidant compounds to reduce the reaction, leading to inhibiting the enzyme activity. However, that would usually result in a change of the sensorial qualities and its nutritional values, which is undesirable. Therefore, in this study, we seek to discover and investigate on the enzyme inhibiting effects of glucose, fructose, sucrose, as well as Maillard products on polyphenol oxidase PPO as well as peroxidase POD. But before we go through the process and results, let us explain the components and what will be involved in this experiment.
Enzymatic browning is an oxidation reaction which is not reversible. It occurs in some of the fruits and vegetables, causing negative effects on color, taste, flavor, and nutritional value. However, it can be prevented by adding some reducing agent to avoid the enzyme to react with the surface of fruits and vegetables. Apple is rich in PPO, polyphenol oxidase, causing it to turn brown so easily. The reaction is a consequence of phenolic compounds oxidation by PPO, which triggers the generation of dark pigments. The active side of PPO contains copper ion, which can change between C+ and C2+. This mechanism lets the enzyme pumps in a lot of oxygen within a short period of time, the polyphenol increasing the rate of browning effect. The enzyme peroxidase allows biological processes to occur. It's an enzyme that oxidizes, that is, break down ester into two alcohols. Glucose is a monosaccharide that provides energy when broken down. It is a polyhydroxyaldehyde with the formula C6H12O6. Although very less amount can be found in glucose solution, the glucose in open chain form potentially acts as reducing agent to prevent enzymatic browning. In this experiment, glucose solutions are investigated of the effect to inactivate enzymatic browning. Fructose has the same molecular formula as glucose, but the way atoms connect is slightly different. It is sweeter than sucrose and it is a reducing sugar. Sucrose is a table sugar we commonly use in kitchen. It is a disaccharide which combines one glucose molecule and one fructose molecule. High concentration of sucrose solution is used as preservative, and in this experiment we'll figure out its effect on preventing enzymatic browning. Maillard reaction, also known as non-enzymatic browning. It is a chemical reaction between an amino acid and a reducing sugar, which require the addition of heat. The reactive carbonyl group of sugar interacts with nucleophilic amino group of the amino acid, resulting in poorly characterized odor and flavor molecules. In this video, we will show you some sugar-reducing agents and their effects on preventing enzymatic browning.
And now, I'm going to describe how the experiment runs. First, carrot and apple are being peeled and cut into 1 cm square cubes. Then, they are blended together and the juice is filtered out with 4 layers of cheesecloth. The juice is added with PPO and POD, which I don't really know where they come from. Then they make sample out of it. After that, different concentrations of glucose, fructose, sucrose, and Maillard products are added in the sample. However, one sample is remained pure as the control group. In the result, the control group is known as 100% relative activity. which means if the result is more than 100%, the enzymes are being activated, and if the enzymes are inhibited, the result will be lower than 100%. Easy to understand, right?
According to our results, the effects of glucose, fructose, and sucrose on PPO is shown in this figure. An inhibiting effect on PPO activity was achieved only at high concentrations of sugar. The result of sucrose stops at 1.1 mole, while glucose and fructose stops at around 2 mole, might be due to the solutions being too saturated. The hypothesis that we predict is that water molecules that are not binded with other substances are free molecule, which is available for chemical reactions. And the higher number of free water molecules there are, the higher water activity it is. This provides a suitable environment for enzymes to work, so the activity of PPO rises. However, the addition of sugar molecules will bind to free water molecules due to the polarity of the sugar molecules. Thus, the environment is in low water activity, causing the enzyme to be slightly inhibited. As for POD, an activation was observed at very low concentrations of glucose and sucrose. The activity remained constant with increasing sugar molarity. Fructose, on the contrary, showed a slight inhibiting effect. Consequently, fructose was shown to be the most effective sugar in inhibiting both enzymes. Maillard product is very effective in inhibiting both PPO and POD. The longer time the glucose or glycine is heated, the more effective it is. It must also be pointed out that PPO and POD activities in the presence of Maillard reaction products caused a lag phase, which increased with increasing volume of the added solutions. In both cases, after the addition of relatively high volumes strongly heated glucose or glycine solutions, no activity was observed for at least 15 minutes. The Maillard products appear to have an important role in preventing enzymatic browning. Their strong anti-browning action could be related to their antioxidant properties, but other mechanisms may be involved. In fact, Maillard reaction products have shown two different effects on PPO and POD: an inhibition of the enzyme activity and the appearance of an initial lag phase. The presence of a lag phase may be because of the reducing properties of some Maillard products, which reduces the early enzymatic oxidation products, preventing the formation of colored compounds. The enzymatic reaction probably starts when all the reducing compounds that are present in the Maillard mixture are consumed. The chelating properties of some Maillard products could also cause a decrease of enzyme activity by reaction with Cu and Fe ions contained in PPO and POD respectively. To conclude our video, it is worth highlighting the fact that fructose and Maillard products falls first and second place respectively as the best possible sugar to inhibit enzymes. And with that fact in mind, we can apply these facts in our actual life applications. And since fructose are relatively easy to get, it is possible to get our hands on one easily. We recommend adding in fructose instead of regular sucrose into your daily dose of fruit juice, which acts as the same function as regular sugar as sweetener in your drink, but even better, inhibiting the enzymatic browning of your fruit juice more effectively, or more than that, to simply rub in fructose to your fruit cuts to prevent browning. So all in all, we hope this video has brought to you good information that is beneficial to you both in your everyday use